Hello and welcome. Today, we're diving deep into the absolutely fascinating world of enzymology. We're going to be exploring the tiny yet incredibly powerful biological machines that literally make life possible. So let's get right into it. So here's where we're headed. First, we'll tackle why enzymes are so vital for, well, the speed of life itself. Then we'll peek under the hood at their structure, see how they grab their targets, and figure out how we can even measure their performance. After that, we'll look at how they're controlled, and we'll wrap it all up with a really cool real-world case study. All right, let's kick things off with a pretty fundamental question. Our bodies are basically just walking, talking bags of chemicals. So why don't we just dissolve? The thing is, all the chemical reactions needed to build you and keep you running are, on their own, painfully, incredibly slow. I mean, really think about that for a second. Digesting your last meal, repairing a cut on your finger, it all happens in the blink of an eye. If you left those same chemicals in a test tube, those reactions might take decades or even centuries. So, what's the secret? How is this possible? Well, the answer is enzymes. These things are nature's miracle workers. They are these amazing protein molecules that act as biological catalysts. What they do is they find and grab onto very specific molecules, which we call substrates, and then they speed up chemical reactions by millions, even billions of times. And the best part? They walk away completely unchanged, ready to do it all over again. They're the ultimate multitaskers. So what is it that gives these proteins these, almost magical abilities? It really all comes down to their incredibly specific and complex 3D shape. Now, for a lot of enzymes, it's not just a simple protein chain. We call the whole working unit a holoenzyme. That's made up of the main protein part, called the apoenzyme, and a little non-protein helper molecule called a cofactor. And that cofactor could be a metal ion or a small organic molecule. And if you look right here, you'll see the most important part of any enzyme, the active site. You can think of this as the business end of the molecule. It's a very precisely shaped pocket or groove where the substrate fits, kind of like a key into a lock, allowing all that chemical magic to happen. And what's so wild is that this active site isn't just one continuous piece of the protein. Look at this diagram of lysozyme. The amino acids that make up this little pocket can actually come from parts of the protein chain that are really, really far apart from each other. The whole protein has to fold up in this incredibly specific way to bring them all together, creating this perfect little microenvironment for the reaction. So because of these unique structures, enzymes have some really key properties. They have extremely high specificity. They only work on one or a few types of substrates. Their catalytic efficiency is just mind-boggling. And they're also pretty sensitive. They're thermolabile, which is a fancy way of saying heat can mess up their delicate shape, and they're also super sensitive to pH. Now, to keep track of them all, we sort enzymes into 6 main classes, based on what they do. Oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. It's like the Dewey Decimal System for biochemistry. Okay, so we know the substrate binds to the active site. But how exactly does that interaction work? Is it a perfect fit from the start, or is it something a little more dynamic? Well, biochemists have come up with 2 key models to explain this perfect handshake. Okay, take a look at these 2 images. On the left, you've got the classic lock and key model. And on the right, you have the more modern induced fit model. Just by looking at the shapes, you can probably already get a sense of the big difference between these 2 ideas. Right, so here's the deal. The lock and key model, which was first proposed by Emil Fischer, basically says that the active site is this rigid structure, a perfect match for the substrate right from the get-go. But the induced fit model from Daniel Koshland suggests something a little more elegant. It says the enzyme is actually flexible. The active site molds and adjusts its shape as the substrate binds, creating an even tighter, more perfect fit. It's less like a key in a lock and more like a firm, welcoming handshake. So now we get how enzymes work. But the next logical question is, how well do they work? How fast can they go? This brings us to the really important field of enzyme kinetics, which is just the study of reaction rates. So, to measure how good an enzyme is at its job, scientists look at what's called the initial velocity, or V0. As you can see on this graph, they measure how fast the product is being made right at the very beginning of the reaction. Why the beginning? Because that's when things are simplest, before the product starts building up or you start running out of substrate. And if you plot that initial velocity against how much substrate you're adding, you get this classic curve. It's called the Michaelis-Menten curve. At first, as you add more substrate, the reaction speeds up, which makes sense. But then it starts to level off. It hits a plateau. Why? Well, it's because all the enzyme's active sites are occupied. They're totally saturated and can't work any faster. Now, since it can be tricky to pinpoint the exact top speed from a curve like this, scientists often use a clever trick called a Lineweaver-Burke plot. It basically turns this curve into a straight line, which makes it way easier to get precise measurements. Now, this curve gives us two super important numbers. The first is Vmax. That's the enzyme's top speed, the absolute maximum rate when it's working as hard as it can. The second is Km, or the Michaelis constant. This is the amount of substrate you need to get the enzyme to work at half its top speed. You can think of Km as a measure of the enzyme's affinity, or its stickiness for its substrate. A really low Km means the enzyme has a high affinity. It's great at grabbing its substrate even when there isn't much around. Of course, in our bodies, it's not always about going at top speed. Biological systems need incredibly precise control. They need ways to hit the gas and ways to slam on the brakes for their enzymes. You know, not all enzymes follow that simple curve we just looked at. There's a special class called allosteric enzymes. They often have multiple parts or subunits, and they produce this characteristic S-shaped or sigmoidal curve. This S shape is a telltale sign of something called cooperativity. It means that when one substrate molecule binds, it actually makes it easier for the next one to bind. This allows for really sensitive on-off switch-like control. Now, one of the most common ways to hit the brakes is through something called inhibition. And this is a great example, competitive inhibition. This is when another molecule, one that looks a whole lot like the real substrate, comes along and binds to the active site. It's basically like someone stealing your reserved parking spot, preventing the actual substrate from getting in and doing its job. And competitive inhibition is just the beginning. With non-competitive inhibition, the inhibitor binds to a completely different spot on the enzyme, which changes its shape and makes the active site less effective. Uncompetitive inhibitors are really sneaky. They wait for the substrate to bind first, and then they lock onto that complex, trapping it. And finally, you have irreversible inhibitors, which form a permanent bond and just shut the enzyme down for good. Think of it like a key breaking off in the lock. Understanding this stuff is absolutely critical because so many of our modern drugs are enzyme inhibitors. Statins, for example, work by competitively inhibiting a key enzyme that your body uses to make cholesterol. Okay, let's put all this theory into practice and look at a really critical, real-world problem: antibiotic resistance. We're going to examine an enzyme called penicillinase, which is the secret weapon produced by dangerous bacteria like Staphylococcus aureus. All right, here's some raw data from a lab experiment. Scientists took this bacterial enzyme and measured how fast it could destroy penicillin at a bunch of different antibiotic concentrations. From these numbers, we can actually calculate the Km and Vmax for this very dangerous enzyme. And when you crunch the numbers, you find that the Km is 5.8 micromolar. Now, that is a very low number, and it tells us something absolutely crucial. This penicillinase enzyme has an incredibly high affinity for penicillin. It is extremely good at finding and grabbing onto the antibiotic molecule. And here is its Vmax, its top speed. This number represents the fastest possible rate at which this single enzyme can just chew through and destroy penicillin. And when you put those two things together, a super high affinity and a really fast reaction rate, you see why this is a superbug's secret weapon. And this is exactly why this kind of analysis is so important. By understanding an enemy enzyme's efficiency, drug designers can work to create new antibiotics or new inhibitors that can outsmart it and give our medicines a fighting chance. So, you see, from the quiet hum of our own metabolism to the frantic arms race of antibiotic resistance, it all comes back to these incredible molecular machines. And their impact goes way beyond biology and medicine. We use them in laundry detergents to break down stains, in making cheese, in brewing beer. The list is endless. So understanding enzymes isn't just about passing a biochemistry exam. It is truly fundamental to understanding and even shaping the world around us and life itself.